All right, let me, before we get started, give a little advertisement for the books, a few books we have on the book table. Three of these I use as part of a class, a course I teach on understanding Darwin, and it's a graduate level course, but I highly recommend these books. One is already 20 years old, by Michael Behe, entitled Darwin's Black Box. And basically, Behe introduces this concept of irreducible complexity that I plan to touch on briefly today. So it's a powerful book. I strongly recommend it, and I think the price is pretty good on that one. Let's see, it's $12.75. Another one is written by a very close friend of mine. He's a pretty well-known geneticist from Cornell, John Sanford. The title of it is Genetic Entropy, and that's going to be one of my main points this morning. I highly recommend that one. And another one is by Stephen Meyer, entitled Darwin's Doubt. It actually is a New York Times bestseller, and it deals a great deal with the fossils and the fossil record, but many other topics as well. So I encourage you, if you're interested, to get that book. And here's this fourth one is just off the press, just published a couple of months ago. It's entitled Contested Bones, and it has to do with the fossil evidence that's claimed to provide some sort of links between ape-like creatures and humans, and so it's an examination of that evidence, and I believe very well done. Okay, so yesterday last evening we talked about language, and then I started a series of two talks on Neo-Darwinism. And so if there are a few of you I know that weren't here last night, let me just go back and summarize at least what we mean by Neo-Darwinism. It was an updated version of Darwinism, what Darwin had published in his 1859 book, except it added some additional features. Neo-Darwin included the laws of inheritance that Gregor Mendel had discovered in the 1860s. And one of the big difficulties Darwin had was he had no idea what the source of variations, genetic variations, could be. And so it was discovered in the early 20th century that mutations existed in the fruit fly lab at Columbia University. And they discovered, for example, that ionizing radiation could modify the genetic makeup of organisms. So the source for genetic novelty, genetic variation in this new theory, this new version of Darwinism, are these random genetic mutations as the source of inherited variation. And then, of course, Darwin's natural selection was a key aspect of Neo-Darwinism. And as I emphasized last night, that viewpoint has essentially continued intact with no modifications since the 1940s. In other words, something on the order of 75 years. That theory has not been modified, and that's essentially currently what the biological community believes is the main driver for diversity and change over the history of life. Okay, so I mentioned that. Well, I summarized 10 reasons. Major problems, things that primarily have been discovered since the 1950s with the advent of molecular biology. And the first major challenge that I covered last night has to do with the fact that the specifications that exist in DNA are linguistic and non-material in their ultimate essence. So a purely materialist hypothesis like Neo-Darwinism offers no explanation for this non-material, this crucial non-material aspect of living systems. So that's in my view, an extremely serious deficiency. One basically renders Neo-Darwinism as a non-starter as far as explaining life. Also last night, we considered the fact that Neo-Darwinism, with these random mutations, is essentially a trial and error search process for genetic improvements. But we saw that the number of trials available, if we consider every organism that's ever lived on Earth as a separate trial, if we assume that each one is genetically distinct, there simply are not enough trials to make any difference in this trial and error search process. The number is so pitifully small, it's not even enough to sample all the possibilities for a single protein with a short protein, 150 amino acids long. And so so that process is dead in the water on that front. So we're going to pick up and look at the other 8 problems that I listed last night. The third one has to do with the cost of substitution of an old trait with a new one via natural selection for a population. And it was found back in the 50s that cost is extreme. And it puts the time span required far beyond the reach of Neo-Darwinism for any process like that to make any difference. And this problem is known as Haldane's dilemma, this extreme cost of replacing an existing genetic trait with a new one. And I mentioned last night that Haldane was one of the founders, one of the 3 founders, along with R.A. Fisher and Sewell Wright, of the field of population genetics back in the late teens and early 1920s. Haldane's book, The Causes of Evolution, published in 1932, represented a major contribution to what came to be known as the Neo-Darwinian synthesis. It reestablished natural selection as the central mechanism of evolution by explaining it supposedly in terms of the mathematical consequences of Mendelian genetics. So Haldane was a key person in the development of Neo-Darwinism. He was a very brilliant mathematician and a very bright guy. And so it wasn't until late in his career in the 1950s that Haldane got around to calculating the cost for fixing a new genetic trait in a population. And he found that this cost of substitution, as he called it, is so high that only one new trait per 300 generations on average could be selected for and be fixed in most higher vertebrate organisms. Just to illustrate that, I'm going to pick one person in the audience here. Well, Carol looked up, so he's a volunteer, and this is I'd say a good thing to be selected for. Now, let's suppose that maybe it's a little late in your life, but suppose that you had a child that had. Well, let's just go back and just let your parents. Your parents had a child, namely you, that had an IQ 50 points higher than average. Okay, that made you very special. All right? Now, let's ask, what would it take for that trait of a 50-point higher IQ to be eventually fixed in the human population so that everybody on Earth, every human being on Earth, had that gene, that new trait that you inherited? What it would mean is that only your offspring could survive in that future population. Everyone else's would have to cease to pass on their genetic makeup. So only descendants of that one individual who inherits this trait can ultimately make up the future population, unless someone happens to marry into that line. Okay, so in this picture, I selected this one guy in the red pullover there, marked with a red arrow. Supposing he was the one with the special trait to be fixed in the future population, what it would mean that everybody else in the stadium, eventually their descendants would have to pass off the scene, no longer leave offspring. So that's a pretty significant price for fixing a new trait in a population. Haldane calculated that for selection to fix that new trait, the number of individuals that are born but must fail to reproduce because they did not inherit the new trait equals about 30 times the population size. Again, this is a huge cost, and it can't be, that kind of cost can't be paid for in one generation. And typically, only about 10% of the population per generation that's about the maximum population can afford to lose, and especially in higher organisms. And so if only 10% are selected away in each generation, that implies you need about 300 generations to fix each new trait. And Haldane realized this was a huge problem. It means that evolution, as he had been advocating, could not proceed at anywhere near the rate that he had assumed. It's a severe limit on the rate of trait substitution. And this problem has become known as Haldane's dilemma, but it has been a trade secret within the population genetics community since soon after Haldane discovered it, because no one was able to come up with a solution for it. So it still is a problem. It's still a trade secret. Most biologists today are unaware of it. So not only most biologists, but certainly most scientists are unaware of it. And certainly most ordinary people have never heard about this glaring problem. But it is a serious problem for evolution. So let's apply this Haldane's result to the hypothesis of human evolution. If we consider a human-like population with a generation time of 20 years, Haldane's estimate implies that in 6 million years, a typical number for the time it's been since we had a common ancestor with some ape-like creature, that there would have been only about 6,000 DNA nucleotide replacements possible. And you get that number by taking 6 million years, dividing by 20 years per generation and 300 generations. That equals 1,000. But there are at least 75 million fixed differences between chimp DNA and human DNA. There's absolutely no way natural selection can account for this difference, even in that standard time scale. So this is a huge problem for evolution. The high cost of fixing new traits. Okay. So it makes it far too slow to work as advertised. Has the population genetics community found a solution during the 60 years since it was first discovered? No, it is simply they've simply sought to keep it hidden, a secret known only in their own circles. So now let's consider the fourth of the 10 challenges. And that is that natural selection cannot even detect, much less eliminate, most bad mutations, and hence cannot prevent genetic decline. So we just to recall, I've already just mentioned these three components of Neo-Darwinism: Mendelian genetics, random genetic mutation, and natural selection. The big problem is that natural selection cannot even see the overwhelming majority of bad mutations, and therefore can do nothing to halt their relentless accumulation. A well-kept secret, even among most professional biologists, is that natural selection has this difficulty. But this was realized by one of the prominent population geneticists of the 1970s. Actually, it was in the 60s. And this Japanese geneticist Kimura, he was keenly aware of Haldane's dilemma and was seeking ways to use the new data from molecular biology to address issues in population genetics. So he was looking at the variation among hemoglobins and cytochrome C proteins in a wide variety of species. And his conclusion is that that selection pressure could not possibly be strong enough to drive the rapid evolution the data seem to imply. And from that, he inferred that most evolution at the molecular level must be the result of random processes like mutation and drift. He spent the rest of his life advancing this idea, which came to be known as the neutral theory of molecular evolution. But as part of that, as he focused on the distribution of mutations and how large their effects were, he established in the population genetics community that most mutations are deleterious, but also have very small effects on overall fitness. So he published plots of this sort, showing the distribution of fitness effect of mutations versus the frequency of such mutations. And he showed the vast majority of mutations have very small effects and fall within what he referred to as this no selection box. And selections within that box were too small for natural selection to detect. And so he pointed out that the bad mutations as well as the good mutations, most of them fall within that box, and natural selection has no ability to do anything about them. And so experts in population genetics have been keenly aware of this reality for several decades. Population geneticists have kept this important reality hidden from the rest of the professional biology community, mainly by reporting the results in terms of sophisticated mathematics in their publications. Most biologists outside the field of population genetics generally are not trained or equipped to understand those mathematical arguments that community uses. Realizing this situation in 2005, Cornell geneticist John Sanford decided it was time for this trade secret of Neo-Darwinism to be exposed so unambiguously that no biologist could possibly question the conclusion that natural selection cannot stop the steady accumulation of bad mutations in a population, and with it be able to stop inevitable genetic decline. So Dr. Sanford's strategy was to develop a simulation tool now known as Mendel's accountant to demonstrate this in a very objective and transparent fashion. So these days, computer simulation provides the means to track hundreds of millions of individual mutations at once and model the mutation selection process with incredible precision. Whereas prior to this, for the most part, population geneticists had used pencil and paper and mathematical formulas to come to their conclusions. What Sanford decided to do was to unleash the power of computers on this problem to be able to do things at a much higher level than had been possible before. And so computer resources available today allow us to do this with a realism hardly imagined even a decade ago. And numerical simulation allows the study of complex interactions of many biological factors simultaneously, for which an analytic solution would be cumbersome, if not impossible. So it's therefore a dramatically more powerful approach for population genetics investigations, because it can deal with multiple variables at once. And actually, John Sanford recruited me. I had recently retired from Los Alamos National Laboratory, and he recruited me because of my computer programming background. So together with another geneticist and another software guy, we put this simulation tool together, now known as Mendel's Accountant. Let me just show an example case. This is a calculation using Mendel that's chosen to approximate human genetic history with the following parameters. This particular example uses a relatively small population size of 1,000. Typically, we use a population size of 10,000 or more, but the results are very similar even with 1,000. For this case, uses a high reproduction rate, 6 offspring per female on average, a mutation rate based on measurements of 100 new mutations per offspring. We use the standard probability type of selection. This particular case, we run it for 200 generations, which we can do just in a few minutes. Use the standard human genome size of 3 billion nucleotides, 23 chromosomes, 1,000 linkage blocks on the chromosomes. We include a significant number of beneficial mutations, at least 2 orders of magnitude beyond what is estimated, just to be generous. In this case, we can, but we don't include recessive mutations. We use a generous fitness heritability factor, and then we have an average mutation effect of about 3 parts in 10,000, so that the average bad mutation reduces fitness by about 3.4 parts out of 10,000. So it's a relatively small effect on average. So, but in this kind of simulation, a crucial issue is the mutation rate. How often do new mutations enter the population? Now, this is based on several fairly recent papers that have sought to measure the mutation rate, and so there are several types of mutations that occur. The simplest one to understand are simply changes in individual letters in the DNA. That's nucleotide substitutions. So it's been found that on average there are about 1 to 300 of those kind of mutations in each child born today. In other words, each child that you had has about 100 to 300 mutations that neither of its parents had. These are new mutations that occur within the germline. Most of them in the male germline. Most of them in the in the cells that produce the sperm in the male. So that's where most of the mutations arise, these new mutations. Then there are some deletions that can occur, but those typically involve many nucleotides, so that there can be as many, between 300 and 3,000 nucleotides affected in these deletions that occur. Then there are also duplications and insertions that have about the same effect. Then there are what are called inversions and translocations, and the exact number is still somewhat unknown, and some conversions. So there's a fairly conservative estimate is that there can be on the order of 1,000 nucleotide differences in the child, that none of which the parents had. But for this calculation, we're only using 100 new mutations per individual. So let's take a look just with this simple example case. And first, let's look at how does the average number of mutations per individual in the population change with time. As you go generation after generation, we're tracking individual mutations, every one of them, with this program. Well, what we find, just in summary, is that the number of mutations, average number, increases steadily proportional to the number of generations. So this is a plot generated by the code, and the red symbols represent the average number of bad mutations. The green, the average number of good mutations, versus the number of generations on the bottom. It's a little hard to read, but 200 generations across the bottom. We have on the left axis number of bad mutations going up to 20,000, and on the right going up to 1.8. These are a scale for the good mutations. So we see that in both cases, in the case of the bad mutations, it's almost a perfect linear trend. Now, if none of the bad mutations were selected away, you have 200 generations and 100 new mutations on average per generation, you'd get 20,000. And it turns out, on average, after 200 generations, the average is 19,802 per individual. And then in the case of the good mutations, you have about 1.8 mutations per individual. If none of those had been selected away, if they had been amplified, you'd had a number greater than 2. Actually, and there's fluctuations. Some of the time it's above the linear trend, other times it's below. But it's following that linear trend very closely. And it's because the fitness effect is small and natural selection can hardly do anything with it, do almost nothing with it. So the mutation counts are very near equal to the mutation rate times the number of generations. This is because the vast majority of mutations have effects too small to be acted upon by natural selection. Selection acts only on the tiny fraction of mutations that exert large effects on fitness. So how does, and I might say we use a distribution of fitness effect similar to what Kimura we saw in that plot by Kimura. It decreases from very small values to larger values in a somewhat of an exponential way. We use a Weibull function, it's called, to describe that distribution. So basically, how does it change with time? Well, if we look at fitness, and that's the next plot, it decreases steadily due to the mounting numbers of bad mutations. And although we don't track it all the way to extinction in this calculation, had we run it a little bit further, we would see that the population would go extinct. There'd be a crash in fitness, dropping it to 0. So in this particular case, after 200 generations, the fitness has dropped by almost 70%, 68% in 200 generations. However, if there were no selection, if there were no bad mutations removed, the population would go extinct in 30 generations. You simply multiply the number of mutations per offspring times the average negative effect, and you get, in 30 generations, 102% fitness reduction. But this natural selection is working, is removing the worst of the bad mutations, and by doing so, the selection reduces the total drop in fitness to only 68% in 200 generations. So this shows that the human race, we might have some little subliminal idea, given how many birth defects there are, that there is a problem. But this quantifies and says there is a significant problem. At the present rate of mutations as measured, the fitness in the human population is decreasing on the order of a percent or so per per generation. Now, back in 1997, a very famous geneticist, James Crow, published this plot. He was concerned about this, that just assuming a 1% decrease per generation in fitness, so that the fitness, the function here is 0.99 to the nth power, where n is the number of generations. He shows that in 300 generations, the fitness drops from 1, a reference starting point of 1, down to under 10% in 300 generations. So that it's simple. It's simple based on the number of mutations that are occurring to realize that there's a big problem as far as genetic deterioration of a population. Okay. These analyses suggest strongly that if presently measured mutation rates have existed during the course of human history, then that human history spans at most a few hundred generations, not thousands of generations. So that means this is one indicator that the human race is most likely very young, hasn't been around that long. It's now clear that natural selection cannot even detect, much less eliminate, most bad mutations. And I didn't mention the real reason for that, and that is that there is noise. There are environmental things going on. Let's just think in terms of plants. Rainfall varies from year to year and place to place, and a genetic type that might do well with the climate of one year might not do so well the next year. So there's this noise going on that obscures these very subtle changes in the fitness. So natural selection just is incapable of seeing these very tiny effects. So again, it's now clear that natural selection cannot even detect, much less eliminate, most bad mutations. Therefore, the combination of natural selection, mutation, and Mendel's law of inheritance, in other words, Neo-Darwinism, leads to genetic decline, not genetic advance. This is precisely the opposite of what the advocates of Neo-Darwinism believe and claim. It's the exact opposite of what we're taught in our biology in high school and in college. Just the opposite. This reality of genetic entropy is something that nobody is talking about today, but is a fatal problem for Neo-Darwinism. The fifth point I want to make here is that biological systems are irreducibly complex, and that was the main point of Behe's book published in 1996, Darwin's Black Box. He does an excellent job at making that case. He points out that Darwin acknowledged that his theory of gradual change by natural selection carried with it a stringent challenge, stating, here's Darwin's quote, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down." So Behe takes up that challenge, and as a molecular biologist, he immediately is able to give many examples of aspects of living systems that could not have come about through this kind of numerous successive slight modifications of a previous system. So he asked in his book, what type of biological system could not be formed by numerous successive slight modifications? His answer, well, for starters, a system that is irreducibly complex. And he defines what he means here. By irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. Okay? So to try to make it as simple to understand as possible, he uses the illustration of a mousetrap. He shows that, he argues that a mousetrap is an example of an irreducibly complex system. Remove any one of its parts and its functionality is destroyed. You take away the holding bar, it doesn't catch a mouse. If you take away the spring or the hammer or the catch or the base platform, all of those are essential if it's going to function to catch a mouse. He says you have to have all those parts together before it has function. So Darwin, as I pointed out last night, had no inkling of what life was like at the molecular level. His knowledge of the small-scale structure of living things was limited to a light microscope, to roughly the wavelength of visible light. Protein machines on which living systems rely are more than 100 times smaller. Last night I quoted the number 500 times smaller, roughly 500 times smaller than what can be seen in an optical microscope. That's where the real action occurs in living systems. So it turns out that thousands upon thousands of features and subsystems within living systems are composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system effectively to cease functioning, and therefore is irreducibly complex. So let's look at a few examples that Behe includes in his book. Now, last night I showed this slide a couple of times, a slide of ATP, the molecule ATP, the primary energy-carrying molecule that powers most cellular processes in all living organisms. And so Behe, in his book, looked at the process involved in synthesizing AMP, adenosine monophosphate. So that's the part of this molecule without the last two phosphate units. Now, he uses this as an illustration of irreducible complexity. The synthesis of AMP, adenosine monophosphate, involves 13 individual separate steps. It requires 12 different enzymes. Each one is a complex protein machine, and it also requires 5 molecules of ATP for energy. So in order to get AMP, you've already have to have ATP. And so the details here are too small for you to see, but this shows these 13 steps and how complex and how difficult it is to synthesize this basic component of every living system. And so he says that there's no way you can put that synthesis process together a single piece at a time. It all has to be there or it's worthless. It's all or nothing. There's no way you're going to synthesize AMP without all of those 12 enzymes and all those 13 steps. So that's an illustration of this irreducible complexity. Other examples he uses include, another one is though he uses systems that involve feedback control, where there is, just like in human systems, complex systems that require things to be stable, it's very common to use what's called feedback control to maintain that stability. And the same is true within living systems. There are thousands upon thousands of feedback control systems, feedback control networks involving usually many enzymes to keep living systems within an acceptable range of stability. And these systems consist of multiple indispensable components and are therefore irreducibly complex. He uses the illustration of blood coagulation, the blood coagulation cascade, it is called. When you cut yourself, this cascade is turned on to create blood clots to stop the bleeding, but that process has to be very carefully controlled, else it will run away and cause all your blood to coagulate. So it has to be very carefully controlled, and once the bleeding is stopped, there has to be a process to dissolve the blood clots. And so all of that is has been worked out and involves this complicated network, which is too faint to see, but is in Behe's book. It's a complex control network. Each of the proteins involved is a complex structure in its own right, with very highly specialized functionality. So living systems have thousands, depending on the complexity of the organism, to hundreds of thousands of these kinds of control networks. The thousands of individual biological machines, commonly constructed from a few protein molecules, individually are also irreducibly complex. He uses the example of the cilium, little structures that move back and forth. For example, that help clear our lungs. In our bronchial tubes, there are millions of cilia that help clear the lungs, and they're little machines that consist of multiple proteins. If all those parts aren't there, those machines simply do not work. So here we saw a related type of machine, kinesin, last night. And another motor protein like kinesin is called dynein, and that's crucial here in this, the motion of the cilium. A sort of a trademark in intelligent design publications. Behe sort of got that started by including the bacterial flagellum as an example of irreducible complexity. The flagella is the means by which many bacteria move around. And basically this is like a motor, electric motor, that causes this flagellum to rotate. And it can change directions within a quarter of a turn. It's an incredible machine built of proteins. So again, without all of these parts working as they're intended to work, that machine has no functionality. Another example of irreducible complexity, the kind that exists in thousands to hundreds of thousands of places in even the simplest living systems. Darwin acknowledged that his theory of gradual change by natural selection carried this stringent challenge, which we've read, that if it could be demonstrated that these systems could not be put together by numerous successive slight modifications, that theory would absolutely break down. Today, hundreds of thousands of glaring examples exist that cause Darwin's theory, and Neo-Darwinism as well, absolutely to break down. Okay, the sixth problem that I have in my list is that favorable mutations are so rare as to be non-existent relative to deleterious mutations. There are good reasons why beneficial mutations are so extremely rare. One is that the DNA specifications generally are for structures that are themselves exceedingly rare. For example, proteins that fold into functional 3D structures. And we saw, let's see, did we see this last night? Yes, that in most cases, most functional proteins have to fold to have functionality. And we saw this work done by Doug Axe back about 15 years ago, where he found he was able, by doing selective mutation on a single protein and looking at how many of those mutations destroyed functionality, he found that the probability of a functional fold occurring in that short protein was 1 out of 10 to the 77th power. So extremely rare. So these are extremely special proteins. And so the idea of being able to make a random change and to get one is essentially impossible. We also saw last night another problem is that linguistic meaning-bearing texts are always highly constrained. Random changes transform such text into gibberish and destroy their meaning. So this was a sequence of 800 random characters with the letter frequencies of the English language, which I wrote a little program to generate these. And this is an example, and so it's just gibberish. And the reason is that there's no meaning in being assigned to those characters, so you wouldn't expect anything but gibberish. But the same thing happens when you randomly change characters in a meaning-bearing sequence. It transforms that sequence into gibberish. So that's another reason why you would not expect random changes to produce beneficial mutations. Another reason is that DNA is polyconstrained due to the fact that the messages that DNA carries are, in many cases, overlapping one another. In other words, the same nucleotides are part of several separate independent messages. And so as I'm going to jump ahead to this next picture. As I was saying last night, in most cases, the complementary strand, say one strand carries a message encoded with the nucleotides, say the specifications for a protein. In most cases, the complementary strand on the opposite backbone, the nucleotides associated with that opposite backbone that are complementary to the ones on the other side, also have a meaning-bearing message. And in addition, it's common for the nucleotides read in the backwards direction also to carry a meaningful message. True also on the complementary strand. And also it's possible to do a frame shift, just shift by one nucleotide and read three nucleotides at a time. After that, you can also, in many cases, get a meaningful message. So you have multiple nucleotide participating in multiple overlapping messages in the DNA. And it's understandable that if a change may not do much damage in one message, but if you've got 3 or 4 or 5 overlapping messages, chances are it can have a major problem, cause a major problem in one or more of the other messages. Okay, so what you have is something like what's called a palindrome. When you have a message that can be read either forwards or backwards. So here's some well-known English palindromes. Abel was I ere I saw Ebla. So that's a message that reads the same either forwards or backwards. Another one is a man, a plan, a canal, Panama. Another one, Madam in Edom, I'm Adam. And another doc note, I dissent. A fast never prevents a fatness. I diet on cod. Okay, you can read that back and it has the same message read forwards and backwards. Another, never odd or even. Rise to vote, sir. In most cases, changing a single letter destroys the palindrome. The fact that DNA code is polyconstrained means that even if a nucleotide change in one of the messages is benign or might provide some degree of positive fine-tuning, it is likely to be deleterious, perhaps in a major way, in the other messages of which it is a part. Therefore, beneficial mutations must be so rare as to be effectively non-existent as far as adding new capabilities to an organism is concerned. And I might just note in passing, people talk about insect resistance or insecticide resistance or resistance to antibiotics that have been identified. But in general, when the effects are examined, what we have is a disabling of an existing functionality that's taking place, not the addition of some new functionality. And when that individual in which that destruction of functionality is put back into the wild, generally it is not as functional, and generally the wild type supplants this one in which the functionality has been disabled. So there's really no evidence. There's almost no evidence, I would say, for any kind of beneficial mutation ever been identified. So the unlimited positive heritable variation Darwin speculated to occur in living organisms, and on which the Neo-Darwinian version of his theory also depends today, is known not to exist. And without this unlimited positive heritable variation, Darwinism in all its forms fail. This is a huge problem, just the lack of positive variation in living systems. Another major problem that's been found is known as the waiting time problem. Even if favorable mutations do occur, the waiting time for even short strings of them to arise is prohibitively long. Let's just say that some new capability, new functionality in an organism requires two simultaneous changes, two nucleotide changes. Let's just think of that, just two nucleotides. They don't even have to be sequential to occur at the same time for that functionality to appear. Like I said, this is known as the waiting time problem, different from, you don't probably have much of this kind of situation in Los Alamos, but in San Diego or LA, it's not, it's probably the norm. Anyway, what if, to be of any benefit, two or more nucleotides must arise together? How long does it take for such a string to arise via random mutation and then to become fixed in a population? There are three reasons it takes a long time. One is that the mutation rate is very low per nucleotide site. If we look at the human genome, the mutation rate per site is low. For a fixed population size of 10,000 and a generation time of 20 years, 4 offspring per couple, some 50,000 years are needed on average for a mutation to occur at a specific site just once in the entire population. Then you can run the numbers. So the mutation rate for a given site is low. Secondly, a desired mutation when it first occurs is frequently lost quickly due to what's called genetic drift and must arise again. So genetic drift is a problem here. For new mutations, single mutations arising, very often they are lost by what's called genetic drift almost immediately in the first generation or second generation. Then requiring, if the sequence becomes, the longer the sequence of new mutations has to be, that number grows with the 4 to the nth power, so it increases strongly with n. All right, so a recent paper focuses on this specific topic of the waiting time problem, and it's in the context of a chimp-like animal, transforming a chimp-like animal into a human. So this paper, I happen to be a co-author, The Waiting Time Problem in a Model Hominin Population. And so we applied our program, Mendel's Accountant, to this question. And here just a summary of the results. Here we are plotting the waiting time in years versus the length of the string that's required. And for a string length of only 2, assuming a huge fitness benefit for the new mutations, the positive mutations, it takes 84 million years for just 2 of them simultaneously to occur, which again makes evolution, makes Neo-Darwinism grind to a halt, makes it impossible for it to accomplish anything. Now here is another plot of this same calculation where we plot it versus fitness benefit. And the waiting time is on the vertical axis, the versus fitness benefit. So the previous calculation I showed was for a 10% fitness benefit. We got 84 million years, but that's unrealistic. Even 1% is unrealistic. It takes 273 million years for that to happen. A more realistic benefit, and it's still large, 1/10% benefit, that the waiting time is almost 1 billion years. So if you're trying to, if you need 2 nucleotides, in other words, and you want to wait until those 2 simultaneously happen in the population, you need to wait for a reasonable type of benefit. You need to wait almost 1 billion years. So that's and actually the secular community has gotten involved in this calculation, and they acknowledge, yes, this is a problem. The study of the waiting time problem demonstrates why appealing to random processes for generating linguistic, meaning-bearing sequences is utterly futile. Again, it demonstrates that non-material linguistic sequences always require an intelligent, non-material source. But going back to the main thing we looked at last night. Because it gives individuals with that 10% benefit a lot bigger advantage. They'll pass that on to their offspring If the benefit isn't very strong, it takes longer for that mutation to be fixed in the population.
Okay, so the 10% benefit is just to the individual?
Yes. No, just to that one individual that has those two mutations. All right, the eighth point, I'm over time, but I'm going to try to run faster here, is the prevalence of what are called orphan genes that shows Darwin's conjecture of common ancestry is now defunct. Okay, what do we mean by common ancestry? Well, it's clearly illustrated by the so-called tree of life, which purports to show that all organisms share a common ancestry. All organisms trace their ancestry back to a simple, generally a single-cell organism. And so this is the concept that we all are descended through a single tree of life back to a single primitive original ancestor. So this was what Darwin drew a simple tree like this. It appears in essentially every biology textbook today. This concept of common ancestry, an essential aspect of Darwinism and Neo-Darwinism. However, the recent ability to sequence the genomes of many different organisms and to compare them with one another has dealt a huge blow to the Darwinian concept of common ancestry. So this ability to actually compare genomes has blown this concept of common ancestry out of the water. And one of the most spectacular ways this has occurred is in the discovery of what are called orphan genes. Orphan gene is a gene that does not match a known gene in any other species. It's a unique gene that occurs only in that species or genus. And here's some quotes. Comparative genome analyses indicate that every taxonomic group so far studied contains 10 to 20% of genes that lack recognizable homologs or similar counterparts in other species. And that was in 2009. More than just orphans. Are taxonomically restricted genes important in evolution? So this is pretty amazing quotes in the literature now. This is one from a paper in 2015 relating to chimps and humans. For the past 20 years, scientists have puzzled over a strange yet ubiquitous genomic phenomenon. In every genome, there are sets of genes which are unique to that particular species. That is, lacking, which means lacking, in other words, genes that are lacking homologs are similar genes in other species. And in this paper, these investigators described 634 orphan genes in humans and 780 in chimpanzees. So there are, if you take the two together, there are more than 1,400 genes that appear in one but not the other. So in the case of humans, there are 634 that, as far as can be determined in the thousands of organisms that have been sequenced so far, don't appear in any other organism. And the same for chimps, and the same for essentially thousands of every organism that's so far been sequenced, they find these orphan genes. And we've just seen the difficulty of getting new proteins or getting new genes, essentially impossible. So where do all these unique genes come from? Well, last night I gave you my answer to that. Here's another paper. I just quote a couple more. A total of 7,184 genes, or 45%, were unique to the Linapithema humili ant, relative to the three other species that they analyzed. So this is an invasive Argentine ant that they've analyzed the genome for, and they find that 45% of the genes in this ant are different from any other ant they've analyzed, or any other organism they've analyzed. Another paper on, this is a leaf cutter ant. We found 9,361 proteins that are unique to this cephalotes, representing over half its predicted proteome. So half the proteins in this ant are unique to that ant, not found in any other. So the proteins coded by orphan genes have invariably found to play essential roles in the organisms in which they occur. So these are not just incidental proteins. In many cases, these are crucial to the existence, functionality of the organism. So why is this discovery of orphan genes so significant? Because it deals a fatal blow to Darwin's idea of universal common ancestry. Basically, it's implying that each species is a unique creation. It has appeared de novo. It is not related ancestrally to any other organism. Else they would share common genes. So that's basically a huge blow, a fatal blow to all forms of Darwinism. So this idea of a tree of life purporting to show all organisms share a common ancestry, that is just, as I said, blown out of the water with the discovery of these orphan genes. This ubiquitous genetic phenomenon, as one of the papers called it, of orphan genes is powerful evidence that each kind of plant and animal at the genus or species level is a separate, near instantaneous, supernatural creation of God, just as Genesis 1 indicates. Okay, so, it's perfectly fine if you want to jump up and shout. All right, I apologize for running over, but hopefully, at least there's a refreshment break here in a few moments. The ninth problem is that the fossil record contradicts the simple to complex and the common ancestry type of history assumed by Neo-Darwinism. First, the staggering numbers of transitional forms required by Darwin's gradualistic hypothesis are systematically missing from the fossil record, and Darwin himself admitted this reality. He said, just in proportion as this process of extermination or extinction has acted on an enormous scale, so must the number of intermediate varieties which have formerly existed on the earth be truly enormous. Why then is not every geological formation and every stratum not full of such intermediate links? Geology does not reveal any such finely graduated organic chain, and this perhaps is the most obvious and serious objection which can be urged against the theory. So Darwin freely admitted that the fossil record does not contain this finely graduated chain of types that his theory would predict. Again, Darwin said, what geological research has not revealed is the former existence of infinitely numerous gradations, as fine as existing varieties, connecting together nearly all existing and extinct species. Now, one of Darwin's contemporaries, the Swiss-born paleontologist Louis Agassiz of Harvard University, who knew the fossil record better than any man alive in that day, emphasized this point strongly to Darwin in his critique of Darwin's book, Origin of Species. Darwin sent him a copy soon after it was written for his critique. So that critique, the infinitely numerous gradations, simply are not there. And that persists as a reality to this very day. This state of affairs led to the emergence of the concept of punctuated equilibrium in the 1970s and 80s through the publications of Stephen Gould, Niles Eldredge, and Stephen Stanley. And this problem, the lack of transitional forms, this is a quote from Stephen Jay Gould: "The extreme rarity of transitional forms in the fossil record persists as the trade secret of paleontology. The absence of fossil evidence for intermediate stages between major transitions in organic design, indeed our inability even in our imagination to construct functional intermediates in many cases, has been a persistent and nagging problem for gradualistic accounts of evolution." So here we have eminent paleontologists acknowledging this. And it led these guys to put forward this idea of punctuated equilibrium. But because they could not provide a plausible mechanism for the sudden appearance of new types of organisms, which their idea required, interest in their idea has faded. But what they pointed out regarding the missing transitional forms is still valid. Again, Darwin was keenly aware of the importance of transitional forms for his theory and the problem of discontinuities in the fossil record that these posed for his theory. He states, "If numerous species belonging to the same genera or family have really started into life at once, this fact would be fatal to the theory of evolution through natural selection." So he admitted that problem. But so why, Agassiz asked, does the fossil record always happen to be incomplete at the nodes connecting Darwin's tree of life? Why have just those transitional types dropped out from the geological record, which would have proved the Darwinian conclusions? So basically the picture has these branches, but no fossils there to demonstrate that they really ever existed. What actually exists and what the punctuated equilibria people pointed out were new types just suddenly appearing in the fossil record and remaining more or less the same until they went extinct. That's why they called it punctuated equilibrium. Equilibrium punctuated at its beginning and often at its end. So this Darwinian expectation simply is not supported by the fossil record. In addition, the Darwinian idea of increasing diversity of life with time is contradicted by the sudden appearance of most animal phyla at the beginning of the record. A couple of good examples of the kind of fossils that they were well known in Darwin's day in the earliest rocks, Cambrian rocks, are brachiopods and trilobites. A huge problem for Darwin. Why are the ancestors for these complex animals nowhere to be found? History according to Darwin's ideas show an increasing branching and increasing diversity with time, whereas the actual record is characterized by striking discontinuity, especially at the very beginning, with a huge variety of body plans appearing in the lowest, most fossil-bearing strata. And this also Agassiz pointed out. He says that the fossil record, particularly the sudden explosive appearance of life in the Cambrian rocks, posed an insuperable difficulty for Darwin's hypothesis. That was some of the feedback that Agassiz gave Darwin. And so here, try to illustrate just how abrupt this sudden appearance is. We have up near the top what's called the Great Unconformity. Below that boundary, we've inverted it as far as the order of sediment is concerned. It's just upside down here. So the earlier In the earlier layers, there are no fossils of multicellular life. And then immediately in the Cambrian rocks, you found 23 out of the 27 of the total animal phyla of the entire fossil record present in the Cambrian rocks. So you have just a sudden appearance of great diversity and also complexity right at the very beginning of the record, just the opposite of what the Darwinian picture would imply. So this is referred to often as the Cambrian explosion. Occurs immediately above a global scale erosional discontinuity known as the Great Unconformity, which is readily visible in the Grand Canyon at the base of the Tapete Sandstone. We'll talk about that this afternoon. Once you start to recognize the layers in the canyon, you can, from the rim of the canyon, trace the Great Unconformity from one horizon to the other. Very clear boundary in the canyon. This sudden appearance of fossils above a global erosional discontinuity is precisely what one should expect from the biblical account of the flood in Noah's day. And from a biblical perspective, it's not at all surprising that the fossil record shows the complexity of life almost as high at the beginning as it is today. But this fact is devastating to the Neo-Darwinian hypothesis. Okay, so the 10th item, and I'm just going to give it very brief treatment, is many diverse lines of evidence indicate that the earth is young, thousands, not billions of years in age. So the vast span of time that Neo-Darwinism requires does not exist, and therefore is a fatal problem for Neo-Darwinism. And some of the, in my view, some of the clearest evidence of this is the spectacular examples of soft tissue preservation, especially those that have come to light in the last 10 years or 15 years. Here are some photographs of flexible blood vessels containing red blood cells found in well-preserved Tyrannosaurus rex bone. So these blood vessels are still flexible. They could be stretched and flexed, and inside you see cells with that are red in color, that are red blood cells without any debate. And in dinosaur bone that's supposedly 65 million years old. Soft tissue simply does not survive for 65 million years in a near-surface environment in a place like Montana. This sort of finding argues strongly that something is profoundly wrong with the radioisotope time scale. Since the next talk addresses multiple evidences that the Earth is young, we'll not go into more detail here. Again, obviously, a young Earth is fatal for Neo-Darwinism. In summary, we have surveyed 10 major ways in which the Neo-Darwinian hypothesis fails scientific scrutiny. What then are the alternatives? Well, like I said, there's only one alternative that I can think of, and that is the near instantaneous supernatural creation of the various kinds of plants and animals we see around us today and that we also see in the fossil record. So thank you for your patience. We'll now take a break, and I will allow some Q&A here, maybe when we come back, or I'll entertain a few questions during the break, if you can catch me. Okay, one quick question.
Behe's idea of complexity, in fact, most of the things you're pointing out here are what I call the straw man. You make up something and say, this is the way it has to happen, and I can show that it doesn't happen. So you make a straw man, and then it's easy to knock it down, and we can all agree. In Behe's complexity, he used a mousetrap. And he had a mousetrap without the little thing that held the spring, right? And he said, see, without that thing, it's not a mousetrap. And a guy in the audience said to him, can I have that? And he said, yes. And he opened it up and made a tie clip out of it. Evolution didn't try to make the mousetrap. It was probably making a tie clip when suddenly something else got in there. And for instance, if you want to look at this, there's a book that you can look at that I think you'll find very readable and very interesting. And you can remember the title because it's called Your Inner Fish.
Okay.
And it shows quite conclusively that sharks have 4 gills. So does a 1-month-old infant.
Now, I take it,
one of those gills turns into your ears.
No, I'd take issue that there's any kind of a gill in a human embryo at any stage.
Well, you can take it, and we can argue that, but it's a very interesting book to read called Your Inner Fish, and it talks about these things. And the key thing was an intermediate form that he was looking for that he actually found. And the point that Neo-Darwinism accepts punctuated equilibrium, it's not a problem for Neo-Darwinism. And then finally, the fact that in the last 30 years, there's just any number of scientists who are saying Neo-Darwinism is a start, but it doesn't do it. The fact of evolution is true.
Well,
the theory of evolution, we're still working on.
I'd say the fact of evolution is a fraud. It's fake science. It's a fraud. It simply is not a fact. There's no way that these complex systems can appear. There's no way you can have.
Well, one last statement. When they were trying to figure out how old the Earth was, they were getting very old numbers in the 1900s. And the top guy on thermodynamics said that can't be true because the only way the sun can be as hot as it is gravitational collapse. And I can show you that gravitational collapse means the sun can only be 100 million years old. It can't be billions of years old. And Henry Norris Russell said the famous thing. He said, I would ask my person if he can't find enough heat in contraction that he go look for a hotter place.
Yeah.
And of course, they found it. So the fact that evolution hasn't been figured out totally is not an argument that it's wrong. It's just an argument that we aren't there. The sun was there.
Yeah.
And
well, I say there's
all the physics said the sun couldn't be there. Yeah. There it was. And when we found out about fusion, then we figured it out.
But I'm saying that the physics that underlie stars and galaxies is trivial, is nothing compared to the complexity that we find in living systems.
And that's why we have the sun figured out and we don't have evolution figured out.
But if you'd heard my first talk last night and my second talk last night, you'd realize what a problem the linguistic specifications are for any kind of evolutionary explanation of life. There's simply no materialist way of obtaining these linguistic structures. So let's break. Let's take a break. Chuck and I can continue. And,
John and I have been doing this for 30 years. We just love each other.
Okay. All right. Thanks.